Welcome to another lecture by MedicoMedics, Cell Biology, Chapter 5, Cell Communication and Adhesion. So in this lecture, we will have an overview of cell junctions, discuss different types. We will talk about the extracellular matrix, integrins and their role, the importance in tissue organization, role in disease, and end with a summary. So cell junctions are specialized structures that connect cells and provide mechanical support and communication. Functions include holding the cells together in tissues, facilitating communication between neighboring cells, and control movement of substances across tissues. So cell junctions are like bridges and tunnels between cells, keeping them connected and working together. Now let's look at types of cell junctions and their specific roles. So we have tight junctions. These seal neighboring cells together to prevent leakage of molecules between them. An example of this is the blood-brain barrier. So as the name implies, it's tight, right? We don't want everything floating around in our blood to enter into our central nervous system. So we have something called the blood-brain barrier, which we will study in other future lectures. Now, we also have gap junctions. These are channels allowing direct communication and passage of ions or small molecules between cells. An example is heart muscle cells for synchronized contractions. And then we have desmosomes. So these provide mechanical strength by anchoring cells together. Example, skin cells to resist tearing. Furthermore, we have adherens junctions connecting actin cytoskeletons of the neighboring cells. An example is tissue remodeling during development. So here you have an illustration of tight junctions. To remember it, just remember the word tight. Another one is gap junctions. And then we have the desmosome. So let's move on to the extracellular matrix. It is a network of proteins and polysaccharides outside cells that provide structural and biochemical support. It's made of collagen, which provides tensile strength. We find elastin, which offers elasticity, and proteoglycans that retain water and provide hydration, and finally fibronectin and laminin. These help cells adhere to the extracellular matrix. What is the function of the extracellular matrix? Well, it organizes cells into tissues. It guides cell migration during wound healing. And it influences cell behavior through signaling pathways. So the extracellular matrix is like glue. And it's like glue for tissues, holding cells in place and helping them communicate. Now let's look at integrins, the proteins that anchor the cells to the extracellular matrix. So integrins are transmembrane proteins that connect the extracellular matrix to the cell's cytoskeleton. Its functions include, indeed, to anchor cells to the matrix. Second, to relay signals between the extracellular matrix and the cell's interior. And it helps in regulating cell behavior, like growth, migration, survival, etc. Why is it clinically relevant? Well, integrins are targets for drugs in diseases like cancer and inflammation. So integrins are like anchors and messengers, holding cells in place and helping them and keeping them informed about their surroundings. Now let's examine how cell adhesion and communication are crucial for tissue organization. So the importance in tissue organization. of these structures. First of all, maintaining tissue structure. So junctions and the extracellular matrix organize cells into functional layers like skin or intestines. Guiding development. So the extracellular matrix directs cell movement during embryogenesis. Its involvement in healing and repair. Cell adhesion helps close wounds by coordinating cell migration. Now, what is its role in disease? Well, if we have a loss of adhesion, so for example, cancer cells lose adhesion to neighboring cells, becoming invasive, so spreading and invading other structures. So we need this adhesion, right? This anchoring to be kept in place. Another one is the extracellular matrix degradation or degradation. So cancer cells, for example, secrete enzymes like MMPs. These break down the extracellular matrix, allowing metastasis or metastasis. And then targeting adhesion in therapy. So we have drugs targeting integrins or the extracellular matrix proteins. These will then be helpful in inhibiting tumor spread. So if we have drugs that can keep this cell anchored to the extracellular matrix, not moving around, it won't be able to go and start invading other structures, leading to chaos, ergo metastasis or indeed cancer. So in summary then, cell junctions ensure structural integrity and communication between cells. We mentioned tight junctions, right? As the name suggests. It's quite difficult getting through. The other one is gap junctions, like in our heart cells. Another one is desmosomes, where cells are kept connected. Then we have the extracellular matrix, which provides supportive structure support and an interactive environment for these cells. We have integrins, which connects the cell to the extracellular matrix and help in relaying signals. If there are disruptions in adhesion, it will contribute to the disease or diseases like, for example, cancer. So, in summary then, cell adhesion and communication are the glue that holds our body together. It keeps our cells connected, informed, and working together in harmony. An easy way to remember these things is to constantly remember what happens if these structures that you are learning now are disrupted. If signals are disrupted, it can overfire, leading to the production of multiple, for example, cell division. So you put this under the umbrella of cancer, and you connect this bit to signaling issues. Another one is the communication between cells, the relaying of signals, which is supported by the extracellular matrix. And these cells are anchored into a place here by the integrins. Play with the idea that these integrins are not functional the way they're supposed to be. So the cell's adhesion to the extracellular matrix will be lost. And indeed, these cells can then move and invade other structures. Again, we are back at cancer. And that's the end of chapter 5. Continue now to chapter 6.